So you've got paper one out of the way. I really hope it went well. Let's now maximise our revision for paper two. So we have a lot of content for paper two, and a lot of it is underpinned by the concepts you have learned for paper one. So you can't forget them. You will need them to apply to your understanding of paper two concepts. So I've put this list together based on my experience of observing common themes that come up on the paper and common areas where answers lack the necessary terminology and linkage of points. So, as we did in our paper one checklist, let's start with electron microscope images. Examiners love these because students struggle with them. So, common images that link to paper two content could be, first of all, sarcomeres, where you have to apply your knowledge of the arrangement of actin and myosin in myofibrils. My biggest piece of advice here is look for two thin dark lines, which are the ends of the sarcomere. These are called Z lines, so helpfully, the end of the sarcomere has been named after the end of the alphabet. The M line will then be in the middle, and it will be surrounded by the H zone, which indicates where there is only myosin. The light band is the I band, and note the vowel I in light, and this is where we have thin actin. Then we have the dark band. And again, to remember that it's the A band, note the A in dark. This is where there is thicker myosin overlapped with actin. Make sure you can compare contracted and relaxed muscles by observing the shortened light band in contracted sarcomeres, where actin has been pulled over myosin by the power strokes. The sarcomere end Z lines will get closer together as the sarcomere shortens. However, the dark A band will stay the same length as myosin length does not change. Now, sarcomeres are the most likely image you will see. However, with photosynthesis and respiration on this paper, make sure you are ready to label or draw the ultrastructure of a mitochondrion or chloroplast. I've tried to pick a few others out which are less likely but not impossible to come up. Here I have a neuromuscular junction where you can see the muscle fibre meeting axon terminals and the vesicles of the neurotransmitter. You might even be able to make out the invaginations of the sarcolemma, which we call the T tubules, and you might also be able to make out the mitochondria, as energy from ATP is needed for neurotransmitter release and repackaging. I've also found an image of a myelinated neuron cross section where you can clearly see the Schwann cell wrapped around the axon. Obviously, this would look different at the nodes of Ranvier, where there are gaps between Schwann cells, where the axon isn't insulated, and action potentials can jump between them by saltatory conduction. My next image is a scanning electron microscope image of a malignant tumour. And we can use this to remind ourselves that malignant cancer cells that are more likely to be life-threatening because the cells can metastasize because they aren't surrounded by a capsule of tissue, they will have a different appearance. The tumours can grow these finger-like projections into surrounding tissue, and whilst I don't have an image of it, malignant cancer cells will also have a larger nucleus under the microscope. We will see a larger, more dense nucleolus, and we would also see a higher percentage of cells with visible chromosomes undergoing mitosis as they are growing rapidly and uncontrolled. Then I've got an image of a ciliated epithelial cell, as the adaptations of these cells are important in the proximal convoluted tubule of the kidney. Remember, they will have finger-like projections to maximize surface area for absorption, selective reabsorption. They'll have lots of mitochondria for active processes and lots of membrane proteins for absorption of molecules. A couple of other kidney images. Don't panic. I think it's unlikely they would show you these, but they are a helpful reminder that there are lots of cells called podocytes with feet called pedicles, which wrap themselves around the capillaries of the glomerulus to increase the surface area for molecules to move across into the filtrate, which is by hydrostatic pressure, not diffusion. We've also got a few pictures reminding us that the capillary has one cell thick endothelium with gaps called fenestrations, which small molecules, not red blood cells or proteins, can be forced out of through the basement membrane of the Bowman's capsule during ultrafiltration. And make sure you refer to it as the basement membrane rather than the filtration membrane as shown on the picture. The final image is of a pancreatic secretory cell. So note lots of vesicles. The pancreas that this is from secretes glucagon and insulin, and this is a big feature of paper 2. So let's move on to our maths equation recall checklist. For paper 2, you should be able to recall all of these equations. And for more information and tips when using the equations, please see my video, Must Memorize Maths Equations. Now, here I've listed the equations you should be able to recall for this paper. For the biodiversity equation, you do not need to recall the equation, but make sure you know that capital N refers to the total number of organisms of all species, and lowercase n refers to the number of each individual species. Now, two common ones for paper 2 are Hardy-Weinberg and mark release recapture. So make sure you can recall these equations, and for both of them, ensure you can list the rules for applying the equations. For mark release recapture, I will reference these a bit later. However, for Hardy-Weinberg, here are the rules. And if the question suggests any selection pressure is occurring, for example, a lethal genotype or a pesticide being used to kill organisms, then you cannot use Hardy-Weinberg to determine allele frequency. Now, remember, P and Q are the allele frequencies. P squared, 2pq, and q squared are the genotype frequencies, and that a dominant phenotype frequency will include p squared and 2pq genotypes. Make sure you convert any frequencies to decimal first. Substitute in what you know to work out which equation you can use first to deduce more information. And for the final answer, double check whether they are looking for a percentage or the number of organisms. Now, whilst we are on maths and inheritance, a common mathematical error in inheritance is to determine the probability of two independent, non-linked phenotypes occurring together. We must multiply the probabilities of the two independent events. I've seen it catch many students out. For example, what is the likelihood of this couple having a girl with cystic fibrosis? You need to multiply the probability of having a girl, 0.5, by the probability of a child having cystic fibrosis. For two heterozygotes, this would be 0.25. So the probability of a girl with cystic fibrosis would be 0.5 times by 0.25, which is 0.125 or 12.5%. Next, let's go through our apparatus and skills tracker from AQA. Overall, at least 15% of the marks for an A-level biology qualification will require the assessment of practical skills. And typically 5 to 6 of these from the AQA list come up every exam season across the papers. So this tracker is a good checklist to go through. For paper 2, I want to mention measuring biomass and energy stored in biomass, as energy in ecosystems comes up on paper 2. We normally use dry mass as it's more representative of the energy stored in the biological molecules. And an important part of the method includes how hot the oven needs to be, 80 degrees, so hot but not hot enough to burn the sample. And doing this until the mass is unchanged. So in practice, your method would have to refer to weighing at regular intervals to determine this. Think about possible control variables such as the known mass, the volume of water that's being heated, the calorimeter itself, as different calorimeters will have different types of insulation to minimize heat loss. Remember, the stirrer and the lid are also key aspects of the apparatus to ensure a valid measurement is obtained, and this is why it's a popular instrument of choice to examine students on. There's also some maths that could be tested where you might be expected to substitute into a given equation. Don't worry, you don't have to recall this one independently. Do note, this is a calorimeter, not a colorimeter. If you have to evaluate this piece of instrumentation, calorimetry often yields a lower estimate of the true chemical energy stored in an ecosystem because heat is transferred to the surrounding air and apparatus rather than just the water, and not all of the biomass burns completely. Next are colorimeters. I still want you to know this and how to make a calibration curve, in particular with reference to CPAC 11 this time. We went through this briefly for the membrane permeability practical for paper 1. You're going to use a colorimeter to measure absorption of light through each known sample. You're going to plot measurements of absorption transmission against known values of our independent variable. Then you're going to draw a line or curve of best fit to produce a calibration curve. Then you can use the calibration curve to determine the glucose concentration of each sample of unknown concentration. The known samples will have undergone the Benedict's test, and therefore a varying concentration of precipitate will be there, which will affect the absorbance or transmission. We can then use our calibration curve to determine the known concentrations by their absorption transmission. Now, another piece of equipment that isn't named in the apparatus checklist but has come up a lot is a respirometer. This has come up on paper 2 or 3 in 2022, 2023, and 2024, so make sure you are familiar with it. Often the rate of respiration in CPAC 9 will have been performed using the rate of decolorization of DC-PIP rather than a respirometer. A version of the equipment setup is shown here, but note it might be done in a water bath to control for temperature, but it would have to be at a temperature safe and ethical for the organisms. You will have your control experiment, which is exactly the same setup but with non-living objects in, and the experimental tube with living organisms placed on a gauze. Now, the gauze is to prevent any harm from the potassium hydroxide, which is in the tube, to absorb any carbon dioxide gas produced during respiration. And this means that as oxygen is absorbed, the pressure in the tube goes down, and the marker, that is often an air bubble in coloured fluid, will move along the lumen of the glass capillary towards the living organism and the area of lower pressure. There's a tap that can be opened to reset the pressure and the position of the bubble marker. And you might be expected to calculate the volume of oxygen taken in, which, if you know the distance on the scale and the radius of the lumen of the capillary, you can substitute into pi r squared times distance. This could also be converted into a rate if you have the time the experiment was done for, which is obviously an important control variable of the investigation, aside from the temperature and the number or mass of organisms.
Remember, it isn't a highly accurate measurement of volume of oxygen used in aerobic respiration, as it is possible oxygen is used in other metabolic processes.
Calculating dilutions or performing serial dilutions is one of the most frequent from this list that comes up in exams. The purpose of a serial dilution is to estimate the concentration of a sample. For example, how many bacteria, but there are too many to count, or they can be used to obtain the desired concentration of a reagent, chemical, or compound. So make sure you can describe how to make a serial dilution in words or calculate volumes needed for a dilution. Now, examiners love a table to complete. Here is an example. And the question is, a scientist wanted to make 50 centimetre cubed of 5 grams per decimetre cube solution of glucose using a stock solution of 40 grams per decimetre cubed glucose. Complete table 1 by giving all headings, units and volumes required to make this solution. Now, in order to work this out, we can use the equation C1 times V1 equals C2 times V2. And with any equations, we always have to substitute in what we know and rearrange the equation for what we don't know. Here we have our known stock solution concentration, our C1, of 40. The desired concentration, C2, is 5, and we want to make 50 centimetre cubed, so V2 is 50. We want to know how much stock solution we add, which is our unknown V1. So we rearrange the equation and substitute in 5 times 50 divided by 40 equals 6.25 centimetre cubed of stock solution. Then we need to add the volume of water, which we can calculate as our desired volume of 50 centimetre cubed minus the volume of stock solution we are adding. So this is what our completed table would look like. There are lots of resources out there with sample questions similar to this if you need more practice. Next on the list is microscopes, scientific drawing and food tests. But my analysis of the papers suggests these are largely paper 1 and occasionally paper 3, but unlikely for paper 2. So come back to these skills for paper 3. Now, separating biological compounds, however, is a big one for paper 2, as CPAC 7 uses thin layer chromatography to separate photosynthetic pigments, and Chapter 21 on gene technology expects us to be able to describe and explain the process of electrophoresis and interpret results. Now, my analysis of past papers shows electrophoresis has come up the last 2 years and chromatography the year before that. So let's give these concepts a quick run through. Make sure you understand why certain steps are important. So I've given a summary here on chromatography. Now, don't get confused with cell fractionation here, where our buffer had to be isotonic to prevent organelle lysis. It had to be cold to prevent enzymes released by cell lysis degrading the organelles. Here we do not need intact organelles. The purpose of the buffer is primarily to maintain a suitable pH and prevent pigment degradation. You will need to recall the RF value equation and explain results. Different photosynthetic pigments absorb different wavelengths of light, allowing plants to absorb a wider range of light energy and increase the rate of photosynthesis. Variation in photosynthetic pigments allows organisms to exploit different ecological niches, and it reduces competition and contribute to natural selection, adaptation, and evolutionary divergence of species. Now, temperature is an important control variable because it affects the movement of the solvent and the solubility of pigments, which may alter the distance travelled and therefore the RF values. Now let's do a quick run through on electrophoresis, and you hopefully recall that it's a process used to separate out substances by their mass and charge, and we frequently see it used in DNA fingerprinting. And it's important to remember that before the DNA fragments are added to the gel, PCR is normally used to amplify the DNA, and restriction endonucleases are used to cut the DNA into fragments at specific base recognition sequences to produce the fragments. The fragments will be of differing size between individuals due to the variable number of tandem repeats in DNA, which tend to be in non coding sequences as it doesn't affect the phenotype. So, DNA is negatively charged and will move towards the positive anode when a current is applied. Now, the reason why the current can flow is there is a buffer, and the buffer contains ions that carry the electrical charge through the gel. And it also maintains a constant pH, which prevents changes in the charge of molecules, such as the DNA or possibly amino acids in a different experiment during electrophoresis. Now, smaller DNA fragments will move further, and the more fragments of a certain size, the thicker the band we will see. On the example here, person A and B are homozygous because they only have one thick band for the section of DNA we are looking at. However, person C is heterozygous because there are two thinner bands, reflecting the two different alleles with different repeat sizes. We could tell if our homozygotes are recessive or dominant if we have allele size information. The larger VNTRs will travel less far, such as for person A, and the smaller VNTRs with smaller fragment size will travel further. Now, if the question includes DNA probes, which are labeled fragments of single stranded DNA with a base sequence complementary to a specific DNA sequence, remember the separated DNA fragments need to be made single stranded before the DNA probes combined. Safe and ethical use of organisms is next on the AQA list, and this has links to CPACs 9 and 10 for paper 2, our rate of respiration practical, and investigating animal responses to different environments. So, handling wood lice safely and ethically could be to use a brush to move the organisms rather than tweezers, and as mentioned before, to use a gauze to prevent wood lice coming into contact with the soda lime in the respirometer practical. Here's a summary of safe and ethical considerations. Next on the AQA list is aseptic techniques. This is more of a paper 1 topic for CPAC 6. However, it is possible that the rate of respiration practical CPAC 9 could be done using bacteria. So just make sure you can recall your aseptic techniques from your paper 1 revision. The dissection skills are also more likely to be assessed in paper 1, so we won't focus on them in this paper 2 revision. So this leaves us with sampling techniques, and this is an important one. It comes up most years across the 3 papers at least once. So let's remind ourselves of the sampling organism summary. Make sure you can select the correct sampling method for the organism. Know the aim of the study. Are you estimating population size of plants or animals? Are you trying to establish a species richness? Or are you looking for a change in frequency as abiotic factors change? In which case you would use a belt or line transect. Now, make sure you can write a method, and this should include variables, equipment, repeats, and data processing. And I remember this using the acronym VERD, VERD. For mark release recapture, you must be able to recall the equation and the rules for the estimate to be valid. And this is one of the reasons why examiners love mark release recapture, because there's maths, there's rules that, if not followed, could lead to invalid estimates. And they love to test your understanding of validity. Next, let's look at our CPAC grid to check these off. Make sure you can identify variables, describe important steps in the method, process and explain results, and evaluate or improve a method. For paper 2, you need to look at CPAC 7 to 12. The next thing to make sure you memorise is the evaluate scaffold. Evaluating experimental data questions expect you to analyse study design and data and present a for and against argument to form a balanced conclusion, or a balanced assessment on the validity of a conclusion the examiners are providing you with. If you've seen my Essential Paper 1 checklist, you will be familiar with this. Now, to help you form your for and against arguments, make sure you annotate key information relevant to the checklist to identify key information in the question. Now, there's a lot on the list, but ultimately you should be asking yourself, what evidence is there for a trend? What evidence is there against a trend? Anomalies, gaps in data, for example. What don't we know? Was there a control, or was there safety information, for example? Also ask, what else could be affecting the dependent variable? A control variable that hasn't been controlled, for example. What else might limit conclusions? Maybe a bias in the sample to males and not females, or the study was in vitro or a mathematical model and not in vivo. I can guarantee you will need to apply your understanding of study design in all papers. Now, I'm going to get you to test your recall here and not show you the answers straight away. So pause the video and see if you can justify these important aspects of validity. Now let's check your wording carefully. Here's a few more questions for you. So pause the video and test yourself on how we obtain reliable data, valid data, accurate data, and reduce uncertainty. Now check your answers again or make a note if you need reminding. Now, the next knowledge check for you, see if you can complete this table to summarise the use of statistical tests. Pause the video again and have a go. Check your answers against the table and double check the wording of your null hypotheses. Now I have a sample question and answer on a conclusion following a statistical test. What is wrong with this answer? So here, the p-value is less than 0.05. So the results are significant and we can reject the null hypothesis. Those parts are correct. However, we can never talk about the results being significant. We have to state the differences are significant when using chi-squared or t-test. And t-test would be the test we are using here because we're comparing means. If we were wording a conclusion on the correlation coefficient, we would have to say the correlation is significant. Now, the second problem with this answer is the statement relating probability to chance. A p-value being less than 0.05 means the probability the differences are due to chance is less than 5%, not more. So that is our essential paper 2 checklist designed to target skills and knowledge with a high likelihood of coming up. If you would like to top up some specific subject knowledge for paper 2, I've made a playlist from my study leave short series specifically for paper 2 content to help you do some quickfire revision based on examiner feedback, mark scheme recipes, and common misconceptions. A new short will appear each day, so keep checking in. If you need to remind yourself on the authorised abbreviations from the official list that AQA released for this exam series, a summary of what is allowed and what common abbreviations we cannot write in our answers is available for free in my SciBrite shop. So you can find the link in the video description. Now, good luck with paper 2. Thank you so much for the kind comments you've taken the time to leave. I really appreciate this as it will increase the visibility of the channel so more students can benefit. I will see you again soon for our essential paper 3 checklist, and please like and share with anyone that might find this video helpful.